Hello, my name is Joshua Mugabe. I welcome you to Reproduction in Living Organisms. Questions 21 to 25. Describe the structure of an ovule and pollen grain in plants. Part B. How do the above mentioned gametes formed? Question 1. Describe the structure of an ovule and pollen grain in plants. The ovule. This contains the female reproductive cells, has a supporting stalk, the funicus, and the nucellus, a mass of cells containing the female gametophyte called the embryo sac. So we have the ovule here. Oh, this is our ovule. It contains the female reproductive cell. It has the stalk, which we call the funicle. It has got a nucellus, where we find the female gametophyte, which we call the embryo sac. And outside it has the chalaza. Then it has the integuments and a small passage we call the micropyle. In angiosperms, those are flowering plants, the nucellus is completely surrounded by a pair of integuments, except at the small pore called the micropyle. In gymnosperms, they have a single integument. So when we look at the ovary, we say that it has got what we call a nucellus, which surrounds the embryo sac. Now, if you find it when it is the nucellus is surrounded by two integuments, a pair, like here we have the integument and then another integument outside, then you know that is a gymnosperm. But if you find the nucellus is surrounded by only one integument, if it has only one integument, then that one is a gymnosperm. Now let's look at the pollen grain. The pollen grain contains the male reproductive cell. It consists of a double wall, a delicate inner wall called the interne, made of cellulose, and a highly sculptured, hard, and resistant outer wall, which is called exine. Inside the pollen grain is a tube nucleus and a generative nucleus. So here we have our pollen grain. When you look at the pollen grain, we say it consists of a double wall. It has an inner wall. The inner wall, which we have called the interne wall, that inner wall. Then it has got a highly sculptured, this one that you see, which is highly sculptured. It is hard and resistant. This outer wall is the one that we call the exine. And inside the pollen grain, there are two nucleus or nuclei. We have one, the tube nuclei, and the generative nucleus. Part B. How do the above mentioned gametes formed? We shall start with the ovule. Formation of the female gametophyte, which is the embryo sac. One of the cells of the nucellus enlarge to form the embryo sac, mother cell. The embryo sac mother cell divides mitotically to give 4 haploid megaspore cells, 3 of which degenerate, and only 1 develops to form the embryo sac. So when you look here, we have the embryo sac of the mother cell. This embryo sac of the mother cell undergoes division, mitotic division, and when it undergoes mitotic division, it forms 4 haploid cells. These are haploid. The embryo sac mother cell is diploid. The megaspore cells that are formed are haploid and 4 in number. Of these 4, only 1 develops to form the embryo sac. And what happens to the 3? They degenerate. The 3 embryo megaspore cells degenerate. Only 1 cell grows to form the embryo sac. The embryo sac nucleus divides mitotically to form 2 nuclei, which migrate to the opposite poles. Each of the 2, either end, divides mitotically twice to form 4 haploid nuclei at each pole. 1 nucleus from either pole moves to the center and fuses to form a diploid primary endosperm nucleus. Remember, we had formed the embryo sac, which is haploid. This one, when it grows, its nucleus undergoes mitotic division to form 2 nuclei, but all of them are haploid. Now these nuclei, they move to opposite poles. And when they move to opposite poles, all of them, they continue to divide mitotically. When they divide mitotically twice, we end up having 4 cells at each pole. and one of the cells from each pore migrate to the center. Those ones that migrate to the center, they fuse. They are cells which are haploid. They fuse. And when they fuse, they form one diploid primary endosperm nucleus. The remaining 6 each get enclosed in a thin cell wall. One of the 3 nuclei near the micropyle forms the egg nucleus. The remaining 2 at the micropyle form the synagogues, which are non-functional ova and do degenerate, while other nuclei at the opposite pole to the micropyle, they become the antipodal cells. So here we have got our embryo sac. We have seen that at the end, there were 4 at each pore, but 2 came in the middle and they fused to form the endosperm nuclei. So meaning that 3 remained on top and 3 remained at the bottom near the micropyle. One of these 3 at the micropyle form what we call the egg nucleus, and the other 2 will turn into what we call the synagogues. Then these ones up opposite the micropyle, they become the antipodal cells. The mature embryo cell thus contains 7 nuclei, 2 synagogues, 3 antipodal cells, and a diploid endosperm nucleus in the center. So we have now here our mature embryo. This is in the middle, the diploid endosperm nucleus, and above we have the 3 antipodal cells. Down we have the ovum or the egg cell and the 2 synagogues. So when you count the total number of nuclei, you will see we have 3 at the bottom and 3 on top, which all of them are haploid, plus 1 in the center, which is a diploid. Then they make a total of 7 nuclei. Part B. We are still looking on how do the above mentioned gametes formed. Now we want to look at the pollen grain. Formation of the pollen grain, repeated mitotic division occur within the sporulonginous tissue, center of the pollen sac, producing a mass of swollen cells called microspore mother cells. Each microspore cell divides mitotically to form a tetrad of haploid microspore pollen cells, which give rise to the pollen grain. So when you look here, you will see we have the pollen mother cell, which is diploid. It undergoes the first meiosis and it forms two microspores cells, which are haploid. And then it undergoes the second meiotic division to form the tetrad of the 4 haploid pollen cells. All of them are haploid. And then these ones, they will secrete cells around themselves, and each one will turn into a pollen grain. These 4 cells, they later separate. Then a single haploid pollen grain nucleus divides mitotically. One of the daughter cells surrounded by denser cytoplasm and forms a generative nucleus, the male gamete, and the other forms the tube nucleus. So when we have our tetrad of the 4 haploid pollen cells formed here, they are going to secrete cells around themselves, and each one becomes a pollen grain of its own. Now, each single pollen grain has one nucleus. This nucleus will undergo mitotic division to form 2 nuclei. One of the nuclei will become the generative nucleus, and then the other nucleus will become the pollen tube nucleus. Each pollen grain develops a hard resistant sculptured outer wall, which we call the exine, and a thinner inner cellulose wall, which we call the intine. So when you look at them, if you see our pollen grain here, the whole of this one is our pollen grain, you can see it has the inner thin wall. That inner thin wall that you are seeing inside, the inner thin wall, is the one that we call the intine thin wall. Then we have that outside layer, which is very hard and sculptured. This membrane here outside is what we call the exine, thick outer sculptured wall. (22) Describe the main events during gametogenesis in male and female humans. Part B, outline the functions of the Sertoli cells. Part A, describe the main event during gametogenesis in males and female humans. Let's start with spermatogenesis. Multiplication phase. At puberty, a diploid germinoepithelial cell, which we call the primordial germ cell of the seminiferous tubule, undergoes repeated mitotic division to form a number of diploid spermatogonia. So we have our diploid germ minoepithelial cell, or what we call the primordial germ cell. This one undergoes mitotic division and produces very many primordial cells, which we call spermatogonia, and all of them are diploid. Then we have the growth phase. Each spermatogonia increases in size and becomes the primary spermatocytes. So when all these spermatogonia increase in size, each of them becomes into what we call the primary spermatocyte. Still, the primary spermatocyte is diploid. Then there is maturation phase. Each primary spermatocyte undergoes the first meiotic division to form two haploid secondary spermatocytes, which undergoes second meiotic division to form four haploid spermatids connected to each other by the cytoplasm. So we have our primary spermatocyte here. It undergoes first meiotic division, and it gives us two haploid secondary spermatocytes. Now, each of these secondary spermatocytes will undergo the second meiotic division. And when they undergo second meiotic division, we end up producing four haploid spermatids, which are connected together by the cytoplasm. Spermiogenesis, in which the spermatids get embedded into the Sertoli cells to be transformed into sperm by losing part of the cytoplasm, condensation of the nucleus into head, and formation of flagellated tail. The mature spermatozoa, the sperms, finally detach from the Sertoli cell and are released into the lumen of the seminiferous tubules. So we have the spermatids. These spermatids, after being formed, they will get embedded or attached to the Sertoli cells. And on the Sertoli cells, they will be transformed into the sperms, which have got a nucleus in the head and a flagellated tail. Now, these sperms that have been finally formed, they will detach from the Sertoli cells and they will be released into the lumen of the seminiferous tubules. Describe the main events during gametogenesis in the male and the female human. We are going now to look at oogenesis. there is the multiplication phase. During the embryonic development, diploid oogonia undergo repeated mitotic division to increase in number. So we have our diploid oogonia here undergoing the multiplication phase. They multiply mitotically and form very many oogonia, which are also diploid. We have the growth phase. Some oogonia undergo mitosis, some of them, to form what we call the primary oocytes, which remain in prophase 1 of meiosis, while the rest, now called follicle cells or granulosa cells, enclose the primary oocytes. So here what we have, we started with one diploid, divided mitotically to form the oogonia, which are still diploid. Some of them, of the oogonia, they formed what we called the primary oocytes. And the rest now, those that did not form the primary oocytes, we now call them follicle cells or granulosa cells. They will go and enclose our primary oocytes. Then we go to the maturation phase. At puberty, granulosa cells multiply to form primary follicle and other cell layers around the primary oocytes. The primary oocyte undergoes meiosis up to metaphase 2 to form the secondary oocyte and the first polar body. So remember, we had our primary oocyte. Now this primary oocyte was surrounded by other oogonia which did not turn into the primary oocyte around to form what we called the primary follicle. Now, the primary oocyte now undergoes meiotic division, the first meiotic division, and it stops at that phase of metaphase 2. And when it stops there, it now forms what we call the secondary oocyte, and also produces one polar body, or the first polar body. The primary follicle develops to form fluid-filled secondary follicle and later Graafian follicle, which encloses the secondary oocyte and the first polar body. Then after fertilization, or at fertilization, the secondary oocytes complete meiosis 2 to form a large otid ovum and a secondary polar body. So we have our secondary oocyte, which is surrounded by what we are calling the Graafian follicle enclosed. It will undergo second meiotic division to complete second meiotic division at fertilization to form a large otite, which we call the ovum, and then the second polar body. The first polar body also undergoes meiosis at the same time to form two smaller polar bodies. All the three polar bodies degenerate and only one function egg remains. So remember, we formed the first polar body, we formed the second polar body. Even the second polar body will also form another polar body, and there will become three polar bodies, which will degenerate. But at the end of it all, we shall remain with only one function egg, which is the ovum. Question 22. Outline the functions of Sertoli cells. The Sertoli cells, they provide support and nourishment to germ cells and the developing spermatids. Secondly, phagocytes, they eat off the cytoplasm of the endospermatids. Thirdly, they secrete a fluid that carries spermatids through the tubules, and they phagocytize foreign particles that invade the tubules. They also secrete androgen binding proteins, which is essential for testosterone activity, particularly on spermatogenesis. They secrete a fluid that releases sperms into the lumen of the seminiferous tubule, spermination. Number 23. Describe fetal circulation before birth. Part B. Describe the developmental changes undergone by the extraembryonic membrane in the human fetus. Describe the fetal circulation before birth. Oxygenated blood from the placenta gets to the liver via the umbilical vein. Part flows through it and the rest bypass it via the ductus venous and gets to the right atrium via the inferior vena cava. So we have the placenta here, where our fetus is going to get its oxygen from. This oxygen will move through the umbilical vein. Some of the oxygen will be taken to the liver, and the other ones will bypass the liver through the ductus venous, and it will go through the inferior vena cava, and it will enter into the right atrium. Largest portion of blood in the right atrium flows through the foramen oval to the left atrium, via then to the left ventricle, via the mitral or bicuspid valves, from which it empties into the aorta that distributes it to the rest of the body. So when our blood comes via the ductus venous and enters into the right atrium, it will enter through the foramen, this gap left between the right atrium and the left atrium. Then it will be pushed to the left ventricle, and from the left ventricle it will be pushed through the mitral valves, or what we call the bicuspid valves. Then it will be emptied into the aorta, and the aorta now will distribute it to all parts of the body. Deoxygenated blood flows through the superior vena cava into the right atrium, where partially mixes with oxygenated blood from the placenta, and via the tricuspid valve gets to the right atrium. So we are having deoxygenated blood coming from the superior vena cava. It will also enter the right atrium. When it enters the right atrium, it will be pushed through the tricuspid valves, and it gets to the right atrium. Here, it is pumped into the pulmonary tract or the pulmonary arteries. Smaller portion passes through the pulmonary circulation via the pulmonary vein, reaches the left atrium. So here what we are seeing, the deoxygenated blood entered into the right atrium. It was pumped into right ventricle. From the right ventricle, it was pumped into the pulmonary arteries, and it was carried through the pulmonary vein. From the pulmonary vein, it entered the left atrium. Due to high pressure in the lungs, a significantly large portion of blood bypasses the pulmonary circulation and goes through the ductus arteriosus, from where it enters the descending aorta directly into the systemic circulation. So this deoxygenated blood, when it comes out of the pulmonary artery and it is taken up, some will go to the lung. But because of the high pressure in the lungs, much of it will bypass through the ductus arteriosus and it will be given to the aorta, where it will be distributed to all parts of the body to join the systemic circulation. Deoxygenated blood then flows through the umbilical arteries back to the placenta for oxygenation, and the cycle repeats. So this deoxygenated blood that has come through the ductus arteriosus, when it connects to the aorta, it will be carried up to the umbilical artery, where it will be taken to the placenta for oxygenation. Part B. Describe the developmental changes undergone by the extraembryonic membrane of the human fetus. The extraembryonic membranes, they include the chorion, yolk sac, amnion, and allantois. Outer cells of the blastocyst, the trophoblasts, they grow and develop into an outer layer or membrane, which we call the chorion, that nourishes and removes waste from the developing embryo. The two cavities appear within the inner cell mass, cells surrounding them, to form two further membranes, the amnion and the yolk sac. The amnion is a thin membrane covering the embryo like an umbrella and offers protection. The yolk sac bears no significant function in humans. The cavity between the amnion and the embryo has an amniotic fluid secreted by the amniotic cells. The fluid supports the embryo and acts as a shock absorber. Cells between the are amnion and the yolk sac form the embryonic disc, which later gives rise to the embryo. The embryonic disc undergoes gastrulation, that is, differentiate into outer ectoderm, middle mesoderm, and the inner endoderm layers. Exchange of materials between the embryo and the mother via the chorionic villi is adequate, but soon the allantois develops from the embryonic hindgut. allantois grows outwards until it gets in contact with the chorion, forming the allantochorion, a structure rich in blood vessels, and contributes to the development of the placenta, a more effective and efficient material exchange structure. (24) Under what circumstances may variation occur in asexual produced individuals? And under what circumstances may variation not occur in sexually produced individual? Part B. State the role of the phytohormones in sex determination of flowering plants. Give an account of the various forms of asexual reproduction. Part A. Under what circumstances may variation occur in asexually produced individuals? In some organisms that reproduce by sporulation, the spores are formed by meiosis. For example, mosses and ferns. Random mutation may also rise and causes variation among asexually reproductive individuals. Environmental factors may make asexually reproduced individuals to vary phenotypically. Under what circumstances may variations not occur in sexually produced individuals? Gametes may be formed mitotically. Secondly, self-fertilization may occur, like in mosses, ferns, and many others. State the role of the phytohormones in sex determination in flowering plants. Gibberellin modifies sex of some flowers by promoting development of male flowers instead of female flowers. Ethylene, on the other hand, promotes the development of female flowers in the place of male flowers. Give an account of the various forms of asexual reproduction. We have fission. It is the division of the cells by mitosis into two or more equal-sized, genetically identical daughter cells. Binary fission, splitting into two, occurs in bacteria, amoeba, paramecium. Multiple fission, schizogamy, splitting into many, occurs in Plasmodium. sporulation, or what we call spore formation. Formation of small unicellular bodies called spore by cell division, which is dispersed from the parent plants and grows into new offsprings under favorable conditions. It occurs in fungi as muca, bacteria, mosses, ferns, liverwood, and algae. Fragmentation. Here the parent organism breaks into two or more genetically identical pieces, each of which is capable of regenerating into a new individual. It can occur in sponges, Spirogyra, Nidalia, flatworms, and many others. Budding. Here the parent cell forms an outgrowth, a small bulge. This increases in size and finally detaches to grow into a new individual. It can take place in fungi such as yeast cells, Nidaria, as Hydra, Obelia, flatworms, tapeworms, and so on. There is vegetative propagation. Here part of the flowering plant with the buds grow and develop into a new plant, at which some stage detaches and form a parent plant and starts to live an independent life. Part of the plant is specialized to give rise to new individuals. For example, the roots, the stems, the leaf are called propagule and act as organs of perennation. Organs of propagation, they include the bulbs, the combs, the rhizomes, the runners, stolons, and tubes. Point 5. How does development of the embryo sac in flowering plants differ from oogenesis in mammals such as humans? B. Compare sexual and asexual reproduction. C. State the adaptations of flowering plants to self-pollination. How does the development of the embryo sac in flowering plants differ from oogenesis in mammals? Let's look at the similarities. Both mitosis is involved. Both results in the formation of offsprings. Differences. Development of the embryo sac, diploid nucleus formed by fusion of two male nuclei. In oogenesis, no diploid nucleus. Embryo sac, 6 haploid cells are formed. Oogenesis, 4 haploid cells are formed. Embryo sac, antipodal cells, synagids, and egg cells are formed. Oogenesis, one egg cell and polar bodies are formed. Embryo sac, only nucleus divides and occurs before fertilization. Oogenesis, the whole cell body divides and occurs during fertilization. Part B. Compare sexual and asexual reproduction. Similarities. In both, there is transmission of genetic material from the parents to their offsprings. Differences. Asexual involves one parent. Sexual involves one or two parents. No gametes in asexual, male and female gametes produced by gametogenesis. Asexual depends on mitosis only. Sexual depends on both mitosis and meiosis, being presented at the same stage of gamete formation. Asexual, offsprings are identical to the parent. Offsprings exhibit genetic variation. Asexual, no zygote formation. Sexual, fertilization to form a diploid zygote. Sexual. Asexual, large number of offsprings are produced. Sexual, few offsprings are produced. Asexual commonly occurs in plants, in simple animals, and microorganisms. Sexual occurs in higher animals and some plants. Asexual, offsprings mature faster. Sexual, offsprings mature slowly. Asexual, population number increases rapidly. Sexual, population number increases slowly. Part C. State the adaptations of flowering plants to self-pollination. The stamen and the carpels, they mature at the same time. Hermaphrodite plants may not open the petals. Hermaphrodite plants may remain underground. Pollen is released onto the stigma by matured anthers before the petals open. Self-compatibility. Pollen is compatible only with the tissues of the style. Flowers are reduced and inconspicuous. Stamens are situated above the stigma, or the anther is close to the stigma. Stigma often coiled to touch the ripe anthers. Style and filament coil on one another. Flowers failing to open, like in Commelina. Thank you very much for participating in reproduction in living organisms. Questions 21 to 25. Please do like, share, and subscribe.